Hello and welcome to the bioenergetics topic. When thinking about exercise physiology, bioenergetics is really important because when we exercise, we need to produce energy at a faster rate to support muscle contraction. So the bioenergetics topic is really about how the muscle produces more energy to support that muscle contraction during exercise. So all of the content that we cover in this topic will be really focused on that very point, and that is, how does the contracting muscle produce more energy to support exercise, different types of exercise, low intensity exercise, or right up to maximal exercise. The first subtopic within the bioenergetics topic is really an overview to set the scene for bioenergetics. So we will cover some background information, and I'll just mention up front, some of this background information, when you first come across it, you may think, what's the point of this? But it will become clear as we add the other layers of the subtopics as to where that background information fits in. So this slide is really a summary slide on energy and human life. So while this slide is not focused on exercise, it gives you an overview of energy transfer. So if we look at this photograph here, it represents foodstuffs. Now, we get our energy from particular foodstuffs, carbohydrates, fats, and the others here, we can also get energy from amino acids. Regarding carbohydrates, glucose is the form of carbohydrate that we use to produce energy in the body. Fatty acids are the source of fats that we use to produce energy. And amino acids are the building blocks of proteins, and individual amino acids can also provide ATP for energy. So, as you're aware, the digestive system is responsible for breaking down these whole foods, and then we have absorption of breakdown products in the digestive system. Then our cells convert the energy stored originally in these foods and convert it into ATP, short for adenosine triphosphate. ATP as an acronym is commonly used, so from this point on we'll just refer to this molecule as ATP. So in terms of exercise, ATP is the source of energy that the muscle uses to contract. So if you think back to skeletal muscle physiology that you will have covered previously, remember that the actin and myosin bind, and there's a power stroke, and multiple power strokes is what causes muscle contraction. So that's where the ATP is used for muscle contraction. Byproducts are carbon dioxide, and you should be aware of the fact that we remove carbon dioxide when we exhale, so the respiratory system is important there, and also water. And given that our bodies are made up of about 60% water, this is not really a waste product, it just becomes part of the water in the body. Now, if we look at the energy stored in these foodstuffs, and when it's transferred into ATP, that conversion process is not efficient, and we produce heat as a byproduct. So if we increase the metabolic rate, so if we go from rest to exercise at a lower intensity to exercise of a higher intensity, we have heat produced in direct proportion to the metabolic rate. So in other words, the higher the exercise intensity, the more heat that we will produce. And you probably all are familiar with the fact that when you do exercise, body temperature goes up, and most often you will start sweating. This is an important point that we will come back to as our topics in exercise physiology progress. If we look at this diagram here, it shows you an ATP molecule. Okay, so remember, it's an acronym for adenosine triphosphate. The adenosine portion is shown here in this part of the diagram, and then you have 3 phosphate groups. Each of these P's represents a phosphate. When the bond shown here between the second and third phosphate group is broken, that releases energy. So this portion of the diagram here, shown in red, represents the energy that's released, and remember, it's that energy that allows the muscle to contract. Now, you'll find throughout these topics I'm often focused on skeletal muscle contraction, because when we exercise, most of the energy that we use or produce above rest is for muscle contraction. Keeping in mind though, at rest we still need energy, and there are multiple processes throughout the whole body, in cells throughout the whole body, that require ATP. But because it's an exercise physiology topic, I will be focused, as I said, mostly on muscle contraction. Okay, so important to note that during heavy exercise, so this means high intensity exercise, so running very fast, swimming long distances at a fairly rapid rate, etc. So the body's total energy expenditure may increase many times above rest. And as I mentioned already, most of the increase is used to provide ATP for contracting skeletal muscle fibres. Just to give you an example, some of that energy would be used for example, an increase in heart rate, but that is cardiac muscle, not skeletal muscle. This is just an overview slide looking at both the formation of ATP and the breakdown of ATP. So try and keep in mind that production of ATP and breakdown of ATP are cycles that are going on constantly in cells throughout the body. When we start exercising and you've got increased skeletal muscle contraction, then the rate of that production and breakdown will increase. So the formation of ATP occurs by combining what we call adenosine diphosphate, or ADP, and inorganic phosphate. The ADP is simply the ATP molecule minus that third phosphate group that was broken when the energy was released. Okay, and the PI, that stands for inorganic phosphate, and again, that represents that third phosphate group that needs to be added back to the ADP, the adenosine diphosphate, short for di-2-phosphates, to form ATP. Okay, so adenosine diphosphate plus inorganic phosphate plus energy forms ATP. Now, a couple of important points. This requires an enzyme. This red text here represents the enzyme ATP synthase that forms the ATP. Now, you've probably heard before that the mitochondria is an organelle within the cell that produces energy. Okay, so most of the ATP that's produced in cells comes from this process here, but there are other mechanisms of ATP production which we will discuss a little later on. So within the mitochondria, you have this process occurring where ATP is produced. Now, the energy that's used to combine the ADP and the inorganic phosphate comes from those foodstuffs that I mentioned on a previous slide. So that's where we get our energy from to reform the ADP within cells. So that's the formation of ATP. This part of the slide refers to the breakdown of ATP. So that's where we break down ATP to access that energy so muscle contraction can take place. The enzyme responsible for breaking down the ATP is ATPase, and the site of that would be at the sarcomere in the muscle where the muscle contraction is taking place. So ATPase would break that second and third phosphate groups apart, so break the bond, combining the second and third phosphate groups. Then you get the formation of ADP and inorganic phosphate, and it's this energy that's then used for muscle contraction. So just to recap, the formation of ATP in the mitochondria uses energy supplied from the foods that I mentioned before. Then when the ATP is broken down in the muscle cell, that energy is released to cause muscle contraction. A really important point to note is that cells don't store ATP. Yes, we can store fat, and I'm sure all of us can grab some kind of subcutaneous fat, so that fat beneath the skin, which is basically an energy store if we need to access it. But we don't store ATP. We can store carbohydrate, and typically we store that in liver and muscle. We don't store protein as such as an energy fuel in the muscle, and we also don't store ATP. So without ATP, being provided constantly by the cell, cells die quickly. As I mentioned before, the fuels to produce ATP in the cells come from glucose, which is a carbohydrate, fatty acids derived from triglycerides, and as I mentioned just a few seconds ago, that's the fat that's stored beneath the skin that we can access if we need to, and amino acids. Now, as we don't store amino acids in the body, we need to access those amino acids from the diet, or if they're not supplied in the diet and we do need to use them, unfortunately we start breaking down body tissues to access those amino acids. For example, muscle tissue. So it's these fuels that are used to synthesize the ATP in cells. You've probably all heard of enzymes, and these are particularly important for the metabolic pathways that produce ATP. What's shown here is an enzyme, and they have a very complex structure. We say they have a tertiary structure and sometimes even 4 levels of structure. And what they do is they increase the rate of chemical reactions. So they don't change the nature of the chemical reaction, but they do speed it up. And really their job is to convert what we call substrate into product. So an enzyme will increase the rate at which substrate is converted into product. If we left the chemical reactions in the body to occur at their own rate without enzymes, it would occur far too slowly to support life. So those enzymes are particularly important. And we have a number of different metabolic pathways that produce ATP. So lots of different enzymes involved in metabolic pathways that convert substrate into product and eventually produce ATP. So enzyme activity is a phrase that will come up quite a bit. When we talk about enzyme activity, we're talking about the rate at which substrate is converted into product. So if an enzyme increases its activity, it means it's working faster and it's causing substrate to be converted into product at a much faster rate, so more product will be produced. So if we're talking about ATP production, in that case, if you increase enzyme activity, you'll increase the rate at which ATP is being produced. Now, we've already mentioned that an increase in exercise intensity, or even going from rest to exercise and then an increase in intensity, will cause an increase in heat production. And remember, that's because the process of producing ATP in the body is not efficient and you have heat produced in direct proportion to the metabolic rate. So in other words, some of that energy that's stored in those foods is lost as heat. If we take a look at this graph, we've got temperature along the x-axis and enzyme activity on the y-axis. And the units for enzyme activity are percentage of maximum. If you think about normal body temperature, it's 37 degrees Celsius, shown here by this line. Now, as we increase our intensity of exercise, we're producing more heat, our body temperature does increase. If we look at this blue line here, it's showing you the effect of temperature on enzyme activity. Okay, so if we go a little bit below body temperature, we'll see enzyme activity is decreased, and if we go above normal body temperature, up to 40 degrees and a little higher, we see an increase in enzyme activity. Now, the phenomenon to explain that is with increased heat, you get increased random movement of molecules, and that increases the chance that the enzyme will collide with its substrate and cause that substrate to be converted into product. So with increased heat, we get increased enzyme activity. Now, what's interesting to see is once we get a little bit above 40 degrees, we see there's a steep decline in enzyme activity. Have a think about that for a couple of seconds and think, why might you get this steep drop-off in enzyme activity? I'm guessing that some of you said that it's because of the enzyme denaturing. and that is the correct answer. So the enzyme, which has a complex structure, as I mentioned, is what we call denatured, and that is when the tertiary structure of the enzyme breaks down. Basically, in simple terms, that means that the characteristic shape of the enzyme, where it functions as like a lock and key to convert substrate into product, that shape has broken down, and that means it's nowhere near as efficient at converting that substrate into product, and that's why the enzyme activity drops off. So a key point from this slide is that as you increase body temperature, you increase enzyme activity. And remember that enzymes are very important for producing ATP, but if our body temperature rises to a certain level, we get a drop-off. And that is very important in terms of heat damage due to exercising, most often in the heat. And we will come back to that later in the topic on exercise in the heat. Okay, so this slide is looking at metabolic pathways that produce ATP. Now, the first thing I should mention is that this diagram here is showing a metabolic pathway. In reality, for most pathways that produce ATP, we have more stages, and typically they're not linear, they tend to be branched. So just keep that in mind, that this is just really a simple example of a metabolic pathway. And because this topic is exercise physiology, we'll look at ATP, so energy, as an end product, and the beginning products can be any of those fuels that allow for the production of ATP. What you can see here, substance 1, 2, 3, 4, and your final product. Okay, so if you remember back to when I said that enzyme activity is the rate at which an enzyme converts substrate into product, you can think of all these substances here as substrates for a particular chemical reaction. So for example, if we said that glucose, being a carbohydrate, was the initial substance in this pathway, glucose would be converted into a product. That product then becomes the substrate for the next chemical reaction, and so on until you reach the final product. Every arrow on this diagram represents a specific enzyme. Okay, so keep in mind that every chemical reaction requires a specific enzyme to catalyze that particular reaction. Enzyme A is shown here as a rate-limiting enzyme. Rate-limiting enzymes typically occur early in a pathway, as shown here, and what they do is they regulate the rate of the metabolic pathway. So in other words, they regulate the speed of the metabolic pathway. So if you think of this pathway like an assembly line in a factory, enzyme A would represent the first person on the assembly line. So if they were to slow down and go very slow, that's going to slow down all these steps ahead of it. If this enzyme speeds up, then it's producing more product and the pathway can increase. Okay, so in general, rate-limiting enzymes control the rate of pathways and typically occur early in the pathway. Another point shown on this slide is that the end product is inhibiting the rate-limiting enzyme. Okay, so end product acts to inhibit, and instead of inhibit, which really sends the idea that it's an on-off switch, think of it more as slows down. Okay, so as you increase the amount of product, you will increase how much this enzyme is slowed down or inhibited. This is a control mechanism that allows the cell, if we're talking about ATP, to produce the amount of ATP that the cell needs. So it's not producing more than it needs, and it's not producing less than it needs. So this is really important in exercise because we want to make sure that our muscle cells are producing the right amount of ATP to support muscle contraction and therefore support exercise. So a metabolic pathway, in summary, is a series of chemical reactions catalyzed by enzymes. And we'll look at specific examples of these metabolic pathways that produce ATP in the following subtopics. And again, important point to note that the product of one enzyme becomes the substrate for the next. So a little bit more on the control of bioenergetics. So what that's really meaning is, how are we controlling the rate of these metabolic pathways? And a key point to start with is the rate limiting enzyme in a pathway and the product. And again, for our example, we'll focus on ATP. It makes sense then that high levels of ATP inhibit the rate limiting enzymes, because remember, we don't store ATP, so the goal of the muscle cell is to produce the right amount of ATP, so in other words what the cell needs at the time. So it makes sense that the higher the ATP that's present in the cell, the more you inhibit this rate limiting enzyme, and that helps to control the ATP flow in the cell. If we look at stimulation of the rate-limiting enzyme, high levels of ADP and inorganic phosphate stimulate the rate-limiting enzymes. And again, this makes sense because these are the breakdown products of ATP. So if levels of ADP and inorganic phosphate are increasing, that is a trigger to the cell that there's not enough ATP there. So this rate-limiting enzyme can increase its speed at which it is converting substrate into product, and therefore this pathway speeds up and ATP production speeds up. There are other factors that control bioenergetic pathways, but the most important are ATP and the breakdown products of ATP, which are ADP and inorganic phosphate. So in summary, ATP will inhibit the rate-limiting enzymes and the breakdown products will stimulate the rate-limiting enzymes. This again is a slide with background information, and this is one of those examples where if you haven't studied chemistry in any detail, or this is rusty, you might think, what's this got to do with bioenergetics? It will become clear in later subtopics why oxidation and reduction are important. So if we start by talking about the hydrogen atom, it's shown here, and the hydrogen atom is made up of one proton and one electron. So it's a very simple atom, however, it's extremely important in energy production. If I link some of the background information we've already covered to this hydrogen atom, remember that foods, carbohydrates, fats, proteins can supply energy or supply ATP. Where this hydrogen comes in is that it's hydrogen atoms that are removed from those foodstuffs. So hydrogen atoms removed from glucose, removed from fatty acids, and removed from amino acids that then allow the cell to use that hydrogen to form ATP. Right, so that's the key point to remember for now, and we will put the pieces together a little bit later on. So two definitions to be familiar with. Oxidation. Oxidation refers to the loss of electrons or hydrogen atoms. Remember, because the hydrogen atom is such a simple atom, if a hydrogen atom is removed from a substance, for example, removed from glucose, we would say that that glucose has become oxidized because it's lost a hydrogen. Reduction is the opposite, and that is when a molecule gains a hydrogen atom or an electron. If we look at this simple diagram here, this is showing you a substance losing an electron and another substance gaining that electron. We say that this substance here has been oxidized and this substance here is reduced. A diagram that's a little more complex over here shows you again a substance that loses these electrons. And when we say loses these electrons, we're really saying that it's losing a hydrogen atom and we're focusing on the electron part, even though there is a proton as well. So substance A is oxidized, it's lost hydrogen, and substance B is reduced because it's gained those electrons. So whenever you have oxidation, where you have a substance losing electrons, you always have that coupled to another substance that is gaining those electrons that have been lost. So as I said, this concept will come back again in following subtopics. NAD and FAD are substances that you'll need to be familiar with. These are acronyms, and because the terms are so long, they're very well accepted to use simply as NAD and FAD, so I don't expect you to know the long versions of the terms. Very simply put, NAD and FAD are carrier molecules that transport hydrogen atoms within the cell. Remember I said on the previous slide that these hydrogen atoms are important because they're removed from, for example, glucose, and then they're used in another part of the cell to actually produce the ATP for muscle contraction. The intermediate step is where NAD and FAD come in that they transport these hydrogen atoms that have been removed from glucose, and then it's used in another part of the cell to produce the ATP. Okay, so the main point from this slide, NAD and FAD, very important molecules in ATP production. They're important because they're carrier molecules that transport hydrogen atoms. You could think of them as hydrogen taxis. NAD and FAD function to pick up hydrogens and then drop them off. Pick them up and drop them off. So these two simple schematics here are showing FAD and NAD with the hydrogens here and here. Okay, so these hydrogens will also then be removed, and then the NAD and FAD will pick up more, and then they'll be removed again. So it's a continuous cycle. It's recommended that you watch the video, it's linked to on LMS, on how the NAD works. So that'll put my explanation on the previous slide into an animation for you. It's now recommended that you attempt the quiz on LMS called Bioenergetics Overview, and that will give you some feedback on the content that you're familiar with in this clip and the content you still need to focus on a little bit. So you can do those quizzes as many times as you like, and then when you're happy with the content in the overview, then go on to the next clip, which is an overview of anaerobic and aerobic metabolism.